Hello everyone. So today we are going to solve two important questions using BLAST, in which we will understand that if we have a given protein sequence or we have a given nucleotide sequence, then how we will find out that uh, to which gene or to which protein does it belong. And we will also solve the related problems like what is the length of that query sequence, which is the organism to which it belongs, what is its e value, etc. So let us come to the problem. So for example, if we have a given protein sequence, this is the protein sequence suppose we have, and we want to find out that this protein sequence belongs to which organism. So we can see that this is an unknown sequence. There is no FASTA format, there is no greater sign, there is no indication in this sequence, there is no accession number. So how we will know that to which protein does this sequence belong? So we have to make the use of BLAST. This is the condition. So BLAST, which is a basic local alignment search tool, and it is from NCBI. So we will go to the BLAST website, NCBI BLAST website, and we will type in Google. We will type NCBI BLAST. So we will open the URL for BLAST. The homepage of BLAST opens. Now in question it is asking that there is the protein sequence, right? And we want to find out to which protein does it belong. So as we know, the purpose of the BLAST is to compare or align the protein sequences itself. So we will go to protein BLAST. So here we will, in the BLAST, we will choose protein BLAST. We will click on protein BLAST. And here it is asking that we can enter query sequence or we can enter accession numbers. So since we are not having the either of them, we are not having the accession number, we are having the query sequence itself. So we will take this query sequence. Now this sequence is not completely in the FASTA format because there is no other information. So simply we will take this query sequence, we will copy it, and simply we will go to the this search box and we will paste it here. This sequence has been pasted here. Now it comes to choose search set database. We want to select the database. So we want to search the protein for this sequence. So what we will do, that the and we also want to find out the organism to which this protein sequence belongs. So we will select from this database protein data bank, not protein data bank. We will select, yes, Swiss prot. So why we will select UniProt Swiss prot? Because it will give us a clear list of the annotated sequences, to which similarly it means that there might be a sequence which is identical or very identical to this sequence, and it is annotated, in which the taxonomy of that organism is given. So here we will select UniProt Swiss prot in order to know the annotation of this sequence. Now we will go down, and in algorithm, since we want to find out the protein, so we will keep selecting BLAST P, that is protein BLAST, and we will run BLAST. So when we ran BLAST, the result will come in few seconds. So we will just go to the question and see what was the question. So the question is that given a protein sequence, the protein sequence is there, we have to find out which protein it is and from which organism does it belong using BLAST program. Then the procedure for this we have done already. Then it is also asking that what is the length of the query sequence, what is the number of the sequences in the databases search, and what is the e-value. So we will just go to the URL, to the website where we will find that whether the result has come or not. So it is still to come. So an important information about this is that we need to search the annotated sequences from from the for a given protein using UniProt or SwissProt. So correspondingly, we will also open the UniProt SwissProt database. SwissProt UniProt database is managed by Swiss Institute of Bioinformatics. So we will go to uniprot.org. It is managed by Swiss Institute of Bioinformatics. So it is also known as UniProt SwissProt database. Now here in UniProt knowledge base, what we need to do is that we need to go to BLAST. Keep opening this. Why we have opened this, I will tell you later. Just we will see for the yes, the result has come here. So when we come down, so here we can see that suppose here these are the results. We want to see the most significant results. So we will select from here 10 so that we can see the most significant results which are identical and similar to the protein sequences we have selected. So here the first result is there. It is for Homo sapiens and the query coverage is 80%. E-value is 1E minus 164, which is less than compared to these below sequences E-value, and the percentage identity is 78.93. It is not 100%, and the accession number is given. Now, whether to know that this particular protein belongs to the protein sequence, we will just what we will do is that we will open this accession number, and when we open this accession number we will see that there is the information about this protein. It is NAD dependent protein deacetylase cytosine 3 mitochondrial. So this is the protein. Now to cross check whether this protein belongs to that sequence, we will take the FASTA format of this sequence. So this is the FASTA format of this sequence, and what we will do that we will copy this FASTA format of the sequence up to here. We will copy it and we will go to the UniProt BLAST page, and here we will paste the FASTA sequence. Now we will run BLAST to understand the complete annotation of this very sequence. So when we run BLAST, it is processing for the information, annotated information about this very sequence, that to which protein does it belong, to which organism does it belong, etc. So meanwhile we will also go to our question, and here it is asking in the question that what is the length of the query sequence. Means the this protein sequence which we have taken, there is we do not know the length of this query sequence. So we will again go to the BLAST NCBI BLAST, where we had opened the detailed result of this protein, and here we want to see its length. So what we will do is we will open its detailed, yes, its detailed information, and here it is said that it is 399 amino acids long. Now here we cross check, the BLAST results are still running. Okay, so it is taking some time. Meanwhile we can see that if this is the protein, it is it was about 78.93, that is 79%, about 79%, it was identical to the protein sequence. So we can check, we are checking for its blast in UniProt for that query sequence. So the result is going to give us, so we can see that this is 399 amino acids long. So it might be, our answer might be that it is the length of the query sequence is 399 amino acids long. So the other is, what is the number of the sequences in the database searched? So the number of sequences in the database searched, it is asking for the number of sequences in NCBI did it search. So since we have taken UniProt Swiss prot database and we had set our algorithm to 100, so it is searching for 100 results. So let us come to the result, this BLASTP result section. So here we can see that the name of the organism, yes, so we can see that about 248 entries are similar to this protein entries to this protein. So the name of the protein here is also NAD dependent protein deacetylase cytosine 3 Homo sapiens. So it is similar to this and how we can see, because the identity is 100%. Here we can see that the identity of this protein is 100% with the sequence. So we got complete clue that the protein sequence belongs to very protein this very protein. Okay, so the answer to our first question is that to which organism does it belong? So it belongs to, we'll see here, it belongs to this, we'll open this, and it belongs to the organism Homo sapiens. So this is the answer. Now the second question is that the length of the amino acid. So length of the amino acid we could find out from here, from NCBI. It is 399 amino acids long. Then the database, how many databases searched. So we also saw that. Now our question is asking that what is the E value. So the E value for this we could see here. The E value was, we will go just back. It was 1E. Yes, we will just run blast once more. Or even we can go back and find out the E value result. Here. Yes. So here the E value for this protein sequence is 1E minus 164. So actually this E value is the expectant value for this protein sequence compared to the protein, which is 1E minus 164. So we got the answers to our first question. Now similarly, there is the second question in which the nucleotide sequence is there. So here it is also asking that we have to find out the details regarding this sequence, that to which organism does it belong, and what is the name of the gene to which this nucleotide sequence belongs, and the accession ID of that particular sequence using BLAST. So again, we will go to the NCBI BLAST page, and here, now since it is a nucleotide sequence, so we will use the BLAST known as nucleotide BLAST. So we will go to the NCBI BLAST homepage. So here we will select nucleotide BLAST because it is a nucleotide sequence and we want to find out the gene. Now what we will do is we will take our sequence from here, the nucleotide sequence. We will copy our nucleotide sequence. We have copied it, and now we will paste it in this search box. We have pasted, and this time the database which we will select will be nucleotide collection only, because our sequence is a nucleotide sequence and we want to search for the gene. So for getting highly specific sequences, highly specific sequences which are nucleotides and the gene name, we will select nucleotide collection database, and we will run blast. So when we run blast, it will the result will take us to the page, to the list of sequences which will be basically the genes correspond to such some genes. So we'll just wait for a few seconds, and we'll go to our question. We'll see that which are the other questions asking in this. So we have to find the details regarding this sequence, and we have to find the organism. Okay, so we'll see how we can find the organism name. So when the details for the details for this very nucleotide sequence will be updated here. So the organism name we can find out from the clicking the details of that ID, accession ID. So it is yet to come. We will see the other sections of the question. So it is asking the name of the organism. Then it is also asking the accession ID, and we are using BLAST. So it is taking some time. Meanwhile, we will go to the UniProt page, and here the information about the proteins. So if we want to cross check the information about the, here we had cross checked the information about the protein sequence, but if we want to cross check the information about the nucleotide sequence, then we can cross check it from GenBank. So now we will keep opening the GenBank if we need it. So we will go to the home page of GenBank. GenBank. It is the page here. We have opened nucleotide, and here we can paste the accession number from our search, and here in the options for submit is also there. So we have to cross check through BLAST. So we will open BLAST. We can open BLAST from here as well. Now we'll just go to our page. We'll see whether results have come or not. So it's just still taking some time. So we'll wait for that moment, and we can see that there are other types of BLAST, like BLAST X, BLAST T, BLAST N. So suppose if our protein sequence were a translated nucleotide, so how we will understand that our protein sequence is a translated nucleotide? Then we'll use BLAST X. Now how we will understand that we want the protein sequence which we want to find out uh, is a translated nucleotide? In that case, the accession number will start with XM. XM it will start. So, and if it is, we will run tblast, and when we have to find the accession number is XM. So in that case, the protein sequences might differ. So it may happen that the protein sequence which we are having, that is having no accession number only, or no, it is not in the form of FASTA format. So in those cases, we will have to use the simply this protein sequence in the using BLAST P only. And if the accession number is given like XM or other, then we will use these, we can use these programs, corresponding programs like BLAST. If XM is there, then it is TBLAST, and if the translated nucleotide is there, then it is BLAST using BLASTX. So suppose this is our result. We've also saved the result here. Yes. So yes, the result has come. So we can see here from this that we want to, suppose we want to get the most significant results. So we will limit it to 10. But here we can see the first result is giving 100% identity. It belongs to mouse. So in our case, this case, the nucleotide sequence which we have taken, that is mus musculus, that is of mouse only. Now the second part of the question says that what is the length of the query sequence? No, what is the accession ID? So we can see that accession ID of this sequence is NM. This is the accession ID. We can open it and we can see the detailed information. So this is the accession ID of that very unknown nucleotide sequence which we have been given. So we are able to get the conclusion from today's video that if we have any protein sequence or we have a nucleotide sequence and we are not knowing its corresponding protein or gene, then we can find it using this method. So in the second case, this nucleotide sequence, the gene which it belongs is this SIR1. This is the name of the gene, and the organism is mus musculus. So hope this video might be helpful for you. So thank you so much. Keep watching my next videos. Thank you.